Hello everyone, it's Mr. Wolf here, and in this video I'm going to be going over formative for experiment C. So the question, does increasing oxygen affect aerobic cellular respiration? So you can kind of see I've drawn my experimental setup here, but let's first just identify our independent and dependent variables. So my question is how does oxygen affect aerobic cellular respiration? So I'm going to be trying, this is my potential cause, so I'm going to be trying to manipulate the amount of oxygen. That's my independent variable. So how are we doing it in this experiment? Well, we have this beaker of hydrogen peroxide in yeast, and we know from our previous unit that when we react hydrogen peroxide with catalase, or almost all organisms have that catalase, it will produce oxygen. So that's given to you. All right? And we could easily take this in and out of the flask. So notice that that little small beaker is just in my flask here, and I could just kind of take tweezers and put it in and out. So specifically, how are we causing there to be more oxygen? We're putting the beaker of yeast in hydrogen peroxide, which will create more oxygen. So that's how we're making more oxygen. Right. What is the dependent variable in this case? Well, I need a way to measure aerobic cellular respiration. So I could either measure maybe the reactants or products, but since we're manipulating the reactants, I can't also measure that as well. So I'm going to have to measure how much CO2 is produced, and that'll give me an idea of if cellular respiration is happening or not. So my dependent variable is carbon dioxide. Specifically, I'm measuring it with the carbon dioxide sensors. All right, and here's another important aspect is, all right, well, what's that measured in? Specifically, where CO2 is measured in ppm, which just means parts per million. All right, so let's look at the whole experimental setup that we have drawn here. All right, so here's normal air, extra oxygen. I have the same amount of P's in each beaker. I decided that I was going to put 15. You could have had a different amount. I said, hey, I'm going to use the same flask. I'm going to collect data for 3 minutes in both trials. The temperature in the room is going to be room temperature, the same, which is about 70 degrees Fahrenheit. And then, and the difference is that with extra oxygen, I have this beaker of yeast and hydrogen peroxide, and these bubbles are supposed to be extra oxygen right here that's produced. All right. And then, again, my sensors right here, these CO2 sensors, are going to be able to measure how much CO2 is in the beaker. All right. So let's look at our constants and controls. Did you include a control group in this experiment? In this experiment, there should be a control group, right? The control group was the normal air because that is the group that's not receiving our treatment, right? Our treatment is adding in this beaker to produce extra oxygen to see if it has an effect on cellular respiration, all right? If you circle no, all right, you could have still gotten by as long as you're explaining why we have a control group. So as long as you're saying, hey, we're comparing the normal air to the extra oxygen, that's the big idea. Like why we have a control group. It allows us to compare one group to the other to see if there's a difference. All right. So then let's go on to constants. Sounds similar, but obviously very different than a control group. Using germinating peas in both beakers, the same amount of peas, same temperature, same type of beaker. These would all be examples of constants that I could use. And then additional parameters. All right. A lot of times I see students writing very vague things like, oh, multiple trials. Cool. How many trials are you going to do? All right. I decided 3. You could have done 4, 5 at a different amount. How many P's are you actually going to put in your chamber? All right. How long are you going to collect data for? Right? I said 3 minutes. You could have said 30 minutes. You could have said 1 minute. It's really up to you. So let's look at the data now. All right. So here's our table. Let's look at that first, zoom in a little bit. And what I'm going to do is cover this up for a specific reason. So this table looks really good. I have my independent variable here, the type of air, and I'm specifying whether it's the normal air or the extra oxygen. I have multiple trials, right, because it asks us for that. And I've been able to take an average. By the way, you may have had a different amount of trials. I even have a title, the effect of oxygen on the rate of cellular respiration. But let's say I put a 4 here. I'm collecting data and I get a 4, right? What does that 4 mean? Right? Well, I don't know. And a lot of times people will put this title in this spot. So you need to have your dependent variable specifically what you're measuring and in what unit you're measuring it. So the amount of carbon dioxide in parts per million. So if I have a 4 here, it's like, 4 parts per million of carbon dioxide. So if I get like a 6 here, 6 parts per million of carbon dioxide. So this is incredibly incredibly important. That's involved in labeling your columns, and I've seen this be missed a decent amount. All right, so let's go on to the graphs, and you'll notice there's actually two different graphs here. Sometimes there's only one type of graph you could have. In this case, there could be two, depending on how you went about it. So let's just start with why you would make a graph, a certain graph. We use a bar graph when the x-axis variable is in categories. So like normal air versus extra oxygen, two different categories, right? But we use a line graph when our x-axis variable is continuous. So for instance, like time is continuous. And so the reason why you might use this, and this is what we'll eventually, how we'll collect data in our experiment, is in class when we looked at the data from the iPad, we saw the amount of oxygen go down over time. The problem is we're comparing two groups, normal air versus extra oxygen, but that's okay. I could just have two different colored lines, and I could still have a line graph here, and I could compare these two just as well. Now I need to make sure that the y-axis is what I'm measuring. It's the amount of carbon dioxide, and again, the units, parts per million, are really, really important. So if we transfer over to the bar graph, you'll notice that I have the same thing. I'm measuring the same thing, amount of CO2, parts per million. All right. Now, I'm guessing most of you made a graph like this because I just didn't give a whole lot of parameters for this one right here. Right? And also, this table would correspond better to this type of graph. All right, so if you did it, again, that's fine. And here I have normal air, extra oxygen on my x-axis. You know, maybe even a better thing to do is to like add in like the air, or I could maybe write condition for a title for my x-axis. But as long as you separate these two things, that's the most important. Right. Now, one thing you'll notice is that I did not include a title for either of my graphs. So you definitely want a title, and it could be something as simple as the effect of oxygen on the rate of cellular respiration, right? Something there, right? The effect of different air conditions on the amount of CO2 produced, something like that. Right. So if you have questions after watching this video, I'm happy to answer them. Maybe write them down on a piece of paper, turn them into the sub, and I'll personally get back to you tomorrow. Thanks for watching.